Hey guys, we're just going to review mitosis and meiosis today, and then there is a mitosis-meiosis review worksheet that you'll do before we move into heredity. So you're welcome to stick with me. I'm going to go through this Google slide as a lesson and explain it. Alternatively, you can also just go, I've posted this in the Google Classroom, the speaker notes that explain pretty much what I'm going to say, so you can also just read through that if that's how you feel like you're going to learn better. Okay, so mitosis and meiosis. These are both forms of cell division, right? So mitosis is just one cell splits into two identical cells. So this is just used for growth. So you're growing. You guys are all probably still getting taller. So your cells are going through mitosis, creating identical cells, and you're growing. Um, Most of the cells in your body are going through mitosis. Meiosis is the creation of sperm and egg cells. So this is important for this unit because we're talking about heredity, right? So we're learning about how you get one chromosome from a sperm and one chromosome from an egg, so that then we see those karyotypes that we were looking at, where you've got two copies of every chromosome, hopefully, although you saw sometimes you can get more than two. Um, yeah, so that's kind of where we're going with this. All right, so the cell cycle. So this is kind of more mitosis stuff. So you've got this red line here, and that is interphase. So interphase isn't part of mitosis. Interphase is just the cell existing and doing like normal cell stuff in your body. So like 90% of the cell's life is spent in interphase. The DNA is being read and turned into proteins, which are doing whatever those proteins do in your body. And then we've got mitosis here um, as the yellow line, and cytokinesis is a blue line. We're going to treat cytokinesis as part of mitosis, so just pretend that that's all one thing there. So mitosis is when your DNA doubles. You have a cell that goes through a series of steps and eventually splits into two cells that are identical to each other. So you're not going to need to memorize like exactly what happens in each phase, but having the basic understanding of what's going on will be important. All right. So we've got interphase here. In interphase, your DNA is in this form called chromatin, where it's kind of just like this ball of spaghetti in your nucleus. So that's really good because when we read DNA, like when it's being read, the genes are being read, you kind of have to have it spread out, right? But then when we actually enter mitosis, we want it to condense into a form that's just easier to move around the cell. So you can see in prophase here, our DNA is condensed into like what you guys saw in those karyotypes. So it's condensed. It's no longer easily readable, so that's why it's not in the same form as it was in interphase. So just before prophase here, our DNA actually doubles. So you get two copies of every chromosome that was in your body in interphase, which means you actually now have four copies of each chromosome. So in your karyotype that you did last week, you saw like chromosome one had two copies. When we start prophase, there's four copies of chromosome one. There's two copies of the egg one and two copies of the sperm one, right? And that's just because at the end of this, we want to have two identical cells. Okay, so prophase, we've got all of the chromatin has condensed. There's two copies of what you originally had. Metaphase, so now you can see we've got this kind of more organized thing happening where everything's lined up at the middle of the cell. So they're also paired. So if you look here, you've got one, two, three, four copies of the chromosomes. And on this side of the cell, on the top here, you're going to have one of the egg ones and one of the sperm ones. Same thing on the bottom. So this would both be like chromosome one, and then this would be like both would be chromosome two. And they're just organizing themselves really well, lining up in the middle, so that we can make sure that when we pull them apart, we end up with the same chromosomes on either end of the cell, so that when it does divide, we have identical cells. And then you can also see these little lines here. Those are spindle fibers. And those are put out by, if you remember the organelle stuff that we talked about earlier, the centrioles. So those send out spindle fibers, and those spindle fibers then, in anaphase, pull those chromosomes apart to opposite ends of the cell. And then telophase cytokinesis here. Now we've got 2 copies of each chromosome, 1 copy from the egg, 1 copy from the sperm, on each end of the cell. They've now uncondensed, and they're in that chromatin spaghetti form again in the nucleus. And the cell is starting to split, and the cell splits into 2. Mitosis is completed. You now have 2 identical cells that are the same as that initial one that you started with. So that's mitosis. These steps in the middle are just really important for making sure that you have the same DNA in both of the end cells there. And there's a video here you guys can watch later. Okay, so some definitions. So these terms are going to come up when we're talking about heredity. So when we're talking about how you get your DNA, your chromosomes from your parents. So we call cells haploid or diploid based on the number of chromosomes in them. So a diploid cell, di for 2, it has 2 copies of every chromosome. So a normal cell that's going through interphase in your body is diploid. It's got 1 egg copy, 1 sperm copy of each chromosome. A haploid cell has 1 copy of every chromosome. So that would not be like a skin cell or a blood cell or something that you had wouldn't be haploid. The only time a cell is going to be haploid is an egg or a sperm. So if you think about it, if your eggs and your sperm had 2 copies of every chromosome, if they were diploid, just like regular cells in your body, when they combined, you would have 2 copies of each chromosome from both of your parents, which isn't how we start off. So we need to have haploid cells as egg and sperm. And then the science term for eggs and sperm are gametes. So gametes are sex cells. They're always going to be haploid cells. And then a somatic cell is a term for all the other cells in your body. So every cell in your body that isn't an egg or a sperm is called a somatic cell. All right, so this is, again, just the stages of mitosis. And we have got a little description on the side here of each of those. I'm not going to go through those again. Here's an actual image. So some of you might have looked at mitosis under a microscope last year. Normally we would do that in the classroom. So this is actually an onion root tip that has been stained, and onion root tips are growing, right? So the cells are undergoing mitosis. So we can actually see really nicely here the different phases of mitosis. So have a look at this, see if you can figure out which one is which. Okay, and answers. So we can see prophase, where we've got our chromatin condensing into chromosomes. Metaphase, everything's lining up nicely on the side of the cell here. We've got anaphase, they're being pulled apart to either side. There isn't really a good telophase that I could find in here, but like telophase cytokinesis would be after anaphase, where everything's being chopped in half. And then here's a cell that's just an interphase, where the DNA is just in chromatin form. All right, so mitosis versus meiosis. So I wanted to start with mitosis because it's a little bit easier to understand the stages. There's a video here that's mitosis versus meiosis, a side-by-side comparison, and I would definitely recommend watching that. It explains it really well. So mitosis and meiosis have similar names, and they do actually have a lot in common. Like they're both forms of cell division, right? But the big picture is mitosis is for growth and repair. Meiosis makes gametes. So mitosis is the division of one cell into two identical, we call them daughter cells. They have the same number of chromosomes as the original cell. Meiosis, one cell actually becomes four cells and they're not identical. And those are gametes, and they only have one copy of every chromosome, so that when they go and fuse with another gamete, they make a cell that has two copies of each chromosome. Okay, meiosis. This is why we started with mitosis. Meiosis has got a lot more stages to it. But if we understand the phases of mitosis, it's not too hard to apply it here. I'm just going to make this a little bigger. So just like big picture, interphase, we've got, two copies of our cells. And then this is just showing from the beginning to the very end. So we've got two copies of a chromosome. At the end of meiosis, we've actually got four cells, and each of them has one copy of that chromosome. And they're all unique. So they're all a little bit different. And then here is just that image split into two so we can see it a little bit closer up. So the first 1, 2, 3, 4, 5 stages here are the same as mitosis. So we've got DNA being copied at the end of interphase. So there's now four copies of each chromosome. They pair up, they line up in the middle, they are pulled apart, and then the cell divides into two. So at the end of that, we've got two daughter cells. One thing that's different here is in prophase 1, so this is going to have two prophases, but in the first prophase, when those chromosomes are next to each other, something called recombination occurs, and some of the genes, some of the alleles, will actually swap between chromosomes. So if you have a look at the chromosomes at the end here, I'll go back to this one. See how some of them have like a little bit of the other color in them. So like let's say this dark blue one is the egg chromosome, and the light blue one is the sperm chromosome. Sometimes we'll end up with a copy of the egg chromosome that's just like as it was. But sometimes there's going to be some crossing over, some recombination, and the chromosome is actually going to look a little bit like a mix of the initial two chromosomes. So that's one difference, kind of right away here, is that recombination. Okay, and then we get down to those two daughter cells. So at this point, it looks very similar to mitosis, but now we're going to go into the second phase of prophase, metaphase, anaphase, and telophase. So in the second prophase, we don't have the DNA double. So we're just going to have one copy of each chromosome. Or sorry, like two copies of each chromosome, like the egg and the sperm one, instead of four, right? And then we're going to go into metaphase. So those are going to line up at the equator. When they're pulled apart, see now how we only have one copy of each one. So like say this is chromosome 1. The one copy is being pulled to one end and one copy is being pulled to the other end. So instead of having two copies of every chromosome, we've only got one when the cell goes through telophase and cytokinesis. And at the end, we have got these four granddaughter cells, each of which has one copy of each chromosome, which is good because then these gametes, these eggs or these sperm, can go on, combine with another one, and end up giving you two copies of every chromosome. Okay, so overview of meiosis, key points. One cell divides into four. Each of them are unique and different. These granddaughter cells are called gametes. They're haploid, so they've got one copy of every chromosome. PMAT is a way that we can remember prophase, metaphase, anaphase, telophase. So just the order of them. So that happens 2 times instead of just once in mitosis. And crossing over or recombination occurs to create diversity in the gametes. So recombination and just having one copy of each chromosome are two ways that siblings can be totally different, even though the eggs and the sperm come from the same two people. Okay, so we've got, yeah, genetically diverse gametes. They combine to create cells with very unique combinations of the parents' genes. So you can have like, I don't know, 10 siblings, and all of them can be very different, just depending on that recombination and which copies and combinations of each chromosome they get. Okay, so that's kind of the big picture of those two. So what you guys are going to do is move on to the Venn diagram worksheet, just compare and contrast the two. Basically, really what I want you guys to get out of this is that mitosis is just growth and repair. One cell becomes two identical cells. Meiosis is to create gametes, sex cells. One egg, or sorry, one cell becomes four non-identical cells, and each of those cells has unique combinations of your chromosomes in them, and just one copy of each chromosome. Yeah, so that's the big picture. Um, read through the speaker notes. Watch this video, the mitosis versus meiosis side by side comparison one. And this video here, mitosis. So both of these are linked in the slideshow. I'd recommend watching both of those. And yeah, hopefully that makes sense. Let me know if you have any questions, and good luck with this.